Welcome to this screencast where I'll be talking about photorespiration and C4 photosynthesis and CAM photosynthesis. So before you look at this, you want to make sure that you know and review and understand the role of rubisco in the light-independent reactions and also the functional anatomy of a leaf. The goal here is to describe and explain what photorespiration is, and then identify and describe the conditions for photorespiration, and then identify and describe some physiological and structural adaptations that plants have to avoid or to limit the negative effects of photorespiration. So in previous screencasts, we had addressed the light-independent reactions, and we had talked about the role of rubisco here in fixing carbon dioxide gas to RUBP to then generate organics. Right? And there happens to be, though, as awesome as rubisco is, there happens to be a problem with rubisco in that rubisco will also bind oxygen. Right? So rubisco will bind CO2 as a substrate, but it will also take oxygen and fix it to RUBP as well. So this creates a bit of a problem because it's going to then limit the number of organics that can be produced. And in that case, your crop yields go down, primary production goes down, and that overall has an effect on the ecosystem as a whole. So photorespiration can be an issue. When would this be an issue? Well, understanding the functional anatomy of the leaf is going to help us out here, and we are looking specifically at here the stomata and the guard cells, part of the leaf. And so when we look at this, understanding the functional anatomy of the leaf and what's going on in the Calvin cycle here, the conditions that are ripe for photorespiration would be living in hot, dry, and sunny environments. So when it is hot and dry and sunny, sunny would be you're going to have high photosynthetic rates. Hot and dry would mean you'd have high transpiration rates. Now, transpiration is the loss of water vapor out of the stomata. So transpiration is losing water out of the stomata. So in a dry environment, you don't want to do that. You want to hold on to as much water as possible. So in a hot, dry environment, the stomata would close. The stomata close. You're like, okay, yay, I'm holding on to water. But the problem there with these closed stomata is that when it's sunny, you're doing photosynthesis. And so your CO2 levels would be going down because you're using carbon dioxide, and your O2 levels would be going up because of the high photosynthetic rates. Now, the stomata being open is a place where CO2 gets into the leaf and where the O2 would come out of the leaf. But with those stomata closed, you're not having this situation going on here. So there's a trade-off, right? There's a trade-off for these plants, is that they hold on to water, limiting transpiration with the closed stomata, but then their CO2 levels continue to go down and O2 levels continue to go up when it's sunny out. Now, this creates a problem because then rubisco begins to preferentially fix oxygen because there's more of it rather than carbon dioxide. So this then leads to less organics or fewer organics being produced. And so why this is called photorespiration is because it's using oxygen here, right? Using oxygen to fix it to RUBP instead of using this carbon dioxide. So this becomes a problem. But there are plants that live in hot, dry, sunny environments, and so they have physiological adaptations as well as structural adaptations to survive in these hot, dry environments and to limit photorespiration. One structural adaptation in these plants is one physiological adaptation. So physiological adaptation, what the cells are doing is they've evolved this C4 photosynthesis. And this involves the cells that are around the vascular tissue here, the veins. And so we're going to zoom in here. We have bundle sheath cells here in dark green, the bundle sheath cells, which is where the Calvin cycle is happening. And then we have these mesophyll cells here where this C4 photosynthesis is going to happen. And there's very limited space between them. In fact, those cells are connected together, and there are these gaps between the cells. If you remember what these gaps in plant cells are called, these are plasmodesmata. Okay. And so these plants that are C4 plants that live in hot, dry environments, they have this other enzyme, which is PEP carboxylase. So PEP carboxylase has a much higher affinity for only CO2. Right? So we can consider this only binding CO2, right? And that's going to fix it to this molecule PEP carboxylase, PEP, which is a 3-carbon. So now if we do the math, 3 carbon plus 1 carbon of carbon dioxide gets me a 4-carbon molecule called oxaloacetate. So this is why this is C4 photosynthesis, because in the mesophyll cells of these C4 plants, they have this extra enzyme, PEP carboxylase, that fixes carbon dioxide to PEP to get a 4-carbon oxaloacetate, and this is why this is C4, because the first molecule that's made there is a 4-carbon molecule. That 4-carbon molecule is then converted in this example into malate, and then that malate, this 4-carbon compound, is delivered right to these bundle sheath cells and is decarboxylated right? And as it's decarboxylated, it generates pyruvate, and then pyruvate can be converted back into PEP, thereby phosphorylating it using ATP. So this is an energetically expensive mechanism. However, it's necessary for survival, right? So this step right here of going from malate to shunting off this carbon dioxide, this delivers more carbon dioxide directly to rubisco right here. So the C4 photosynthesis is a way to fix carbon dioxide, right, from the atmosphere, right, and feed it directly to rubisco. Right? So we're going to transfer carbon dioxide, a CO2, in the form of a carboxyl group right directly to the 5-carbon RUBP here, right, and feed it right to rubisco. So there's no gaseous part to this. Right? So we're sort of supercharging the rubisco here. And then the Calvin cycle can continue spinning around, and as it does so, then we yield sugars or organics, which get into the phloem, etc. So this C4 photosynthesis is an adaptation to keep photorespiration down, to limit photorespiration. A note here that C4 plants, right, also do C3 photosynthesis, that is, also do the Calvin cycle. Right? And that is the way that they make the sugars. This is the way that they make the organics, is using that Calvin cycle. But the C4 is added, right, as a way to get CO2 directly to rubisco. Directly. Right? And remember, why we need to do this is because the CO2 gas levels are really low because the stomata are closed. So another physiological adaptation are the crassulacean acid metabolism, or CAM plants here. And these CAM plants will do something like the C4 plants over here. So again, this carbon dioxide is fixed into organic acids like oxaloacetate, and then the CO2 is delivered directly to the Calvin cycle. In a CAM plant, it's very similar, but at nighttime, when these stomata are open because it's cooler, it's not as dry, it's not as hot, you're not losing as much water vapor. So in this case, CO2 can come in, and CO2 will be fixed by PEP carboxylase, and then the organic acids get stored in the cells so that during the daytime, when the Calvin cycle is running more vigorously because you're doing photosynthesis, you're doing the light-dependent reactions, you're getting more ATP, you're getting more NADPH to run this Calvin cycle. As this Calvin cycle begins to run, you start to churn out sugars there. So then these organic acids are decarboxylated directly to this. And so this wouldn't be like first thing in the morning. This would be like, as the day goes on, midday, late day, where you're going to have these organic acids then broken down, decarboxylated, and regenerated back up here. So these CAM plants are doing something very similar to the C4. It's just that they're storing these organic acids for during the daytime, whereas this is happening usually just during the daytime here for the C4 plants. This is running real time. Okay, so just variation on a theme there between the C4 and the CAM. What are these plants that do this C4 and CAM? You have these succulents like pineapples. You have grasses like sugar cane. Right? These are big-time cash crops, so it behooves them to, they live in hot, dry environments and have these adaptations for survival. One other adaptation in plants that live in hot, dry, windy environments. Right? So not only hot and dry, but also windy. If it's windy, this tends to dry out the air. And so if you look microscopically, structurally, at the leaves of these, so here's a cross-section through the leaf. Right? First, they have a very thick upper epidermis and thick waxy cuticle. Right? This is so these are structural adaptations, so thick waxy cuticle, right? And a thick upper epidermis. The palisades parenchyma, the spongy parenchyma is here. And what you notice is that there are these little, on the lower epidermis here, there are these recessed pits. And so this is a recessed pit. And what you see is that these are guard cells, guard cells, guard cells. So the stomata here are in this little alcove, this little recessed pit. And they have these little hairs that come off here, though. So these are all extensions of the cytoplasm, little hairs in here. And so as it's windy, as the wind is going this way, right, drying out the air over here, right, drying out the air down here, the air up in here maintains a little bit of water vapor. The moisture is in there. It's not as dry. And so the stomata then are able to remain a little bit open. So this is kind of like a little harbor, right, away from where the wind is. And this is a Nerium oleander leaf here. And so this is a cross-section through this oleander leaf, right? So a little bit about photorespiration there and the issues associated with it. You should be able to identify and describe the conditions that photorespiration would take place under and some physiological and structural adaptations to photorespiration and for survival in hot, dry, sunny, windy environments. Okay, thanks for watching.